Hi guys, this is Science Simplified and welcome to the video about cells. So you might ask, what are the differences between animal and plant cells? Look at the two diagrams that are being drawn. Can you see any differences? Stay till the end of the video to find out.
Now moving on to this lesson's main objectives, which are learning the microscopes that are used to magnify the cells, finding the parts of the plant and animal cells and differentiation. In microscopes there are two types: light and electron microscopes. Light microscopes magnifies the object up to 1000 times and comes out as colored images, so you can see a simple microscope being drawn. Now, let's find out the parts. This is the eyepiece where you can attach the lenses. This is the coarse focusing or adjustment knob. And below that, is the fine focusing or adjustment knob. This is the neck of the microscope. And these are the objective lenses where you can attach times 4, 10 and 40. And just below it is the stage of the microscope. Moving on to the electron microscope, which magnifies the object up to more than 200,000 times and are black and white but can be colorized. Now let's jump into the animal cell. Take a look at the cell being drawn here. The cell membrane is a partially permeable membrane which controls the substances entering and leaving the cell. The nucleus consists of nuclear envelope, nucleoplasm, nucleolus and chromatin. Now look at the diagram that is being drawn here. The nuclear envelope separates the contents of the nucleus from the rest of the cytoplasm. The nucleoplasm is the dense material within the nucleus. The nucleolus plays a part in making proteins in the cell. And chromatin is a mass of long thread-like substances in which each of them are made out of proteins and compounds called deoxyribonucleic acid and carries instruction that the cell needs to carry out its activities. During cell division, the chromatin condenses to form chromosomes. Moving on to the mitochondria, which is an organelle bounded by two membranes and provides energy to break down the food substances by oxidation during cell respiration. The energy is used to perform cell activities like growth and reproduction. This is a diagram of a rough endoplasmic reticulum. It consists of a network of flattened spaces lined with membrane. Ribosomes are attached to its outer surface, which involves in protein synthesis. Its outer surface is continuous with the nuclear envelope. So what are ribosomes? They are small round structures, either attached to the membrane or RER, or lie freely in the cytoplasm. It is attached to RER to make protein to be transported out of the cell, or they can lie freely in the cytoplasm to make proteins that are used within the cell. Take a look at the diagram of the smooth endoplasmic reticulum. It has no ribosomes attached to its membranes. It synthesizes substances such as fats and steroids and converts harmful substances to harmless substances known as detoxification. The Golgi apparatus chemically modifies substances made from the RER and stores and packages these substances in vesicles for secretion out of the cell. Now let's move on to plant cells. This is a diagram of a plant cell. In the plant cell there is chloroplast, which contains a green pigment called chlorophyll, which is essential for photosynthesis. There is also cell wall, made by cellulose and found in only plant cells. It protects the cell and gives it a fixed shape. It is also fully permeable. In animals, the vacuoles contains water and food substances and exists temporarily. While in plants there is a single, large and central vacuole that contains cell sap. Moving to differentiation. Differentiation is the process where new cells develop special structures or lose certain features to enable them to carry out certain features. Example 1 is red blood cells. As you can see from the diagram, it has a circular, biconcave shape to increase surface area to volume ration, and it has no nucleus in order to store more hemoglobin. It is elastic so that it is able to squeeze through narrow capillaries. Example 2 is the root hair cells. From the diagram, it has a long narrow protrusion, increasing the surface area to volume ratio for faster rate of absorption. It also has a large vacuole with concentrated salt solution, also causing faster rate of absorption.